Hello, everyone, and welcome to today's live broadcast, introducing an ancestrally diverse whole genome data set for select cohort and control research applications presented by Benjamin D. Solomon, MD. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN Bioinformatics. QIAGEN offers a targeted product portfolio that enables insights from samples for virtually any application involving DNA and RNA, the building blocks of life. QIAGEN has partnered with the Inova Translational Medicine Institute to offer a collection of high-quality annotated whole genome sequences for diverse clinical applications. To learn more about QIAGEN Bioinformatics, please visit www.qiagenbioinformatics.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the right side. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Benjamin D. Solomon, Chief of the Division of Medical Genomics at the Inova Translational Medicine Institute. Dr. Solomon is an accomplished scientist and medical geneticist who is dual board certified in pediatrics and clinical genetics through the National Human Genome Research Institute. He has previously held positions at the National Institutes of Health, researching the genetic and genomic causes of both rare and common conditions, especially certain types of congenital anomalies. At the Inova Translational Medicine Institute, Dr. Solomon leads the Medical Genomics Division, a group of clinicians and researchers that focuses on providing genetic and genomic medical care, discovering new explanations for genetic disorders, and studying the best ways to integrate cutting-edge genomic resources into clinical practice. Today, he will be speaking about groundbreaking studies that have generated a data set of nearly 7,000 fully sequenced genomes annotated with familial relationships, ancestral origin, and clinical phenotypes that have been the focus of a number of recent publications. Please join me in welcoming Dr. Solomon. I will now turn the presentation over to him.
Thanks very much for the kind introduction. I trust that folks out there will let me know if they're having trouble hearing me or if the connection's not good. Excuse me, but I'm going to go ahead and dive right into the presentation. Again, I'm very grateful for the opportunity to present what we're doing and what we hope to do and how we've partnered with QIAGEN and Ingenuity for some of these projects. By way of disclosures, I want to mention one thing. I grew up for most of my childhood in New York, so perhaps as a consequence, I tend to talk very fast, especially when I start to talk about something I'm excited about, such as genetics and genomics and the clinical applications. So I'll also ask that if I'm speaking too quick for human intelligibility, please someone shout out or send me a message so I can slow down a little bit. And with that, I'm going to go ahead and dive right in. The talk is kind of loosely organized into three overlapping sections. First, I'm going to talk about background, about who we are and what our studies entail, and the types of data that we're generating. Second, I'll describe examples of some of the cases that we focus on, and I've chosen intentionally some of the Mendelian type or perhaps N of 1 type of analyses. Though I'm not going to go into these in the talk, we separately do a lot of work on complex type diseases such as preterm birth and other conditions that are multifactorial. And if folks have questions about some of the non-N of 1, some of the more sophisticated and long-term analyses that we do, I encourage them to contact me or discuss them further if any questions, ideas for collaborations, or other input arises. And then finally, if you'll indulge me at the end, I'm going to talk a little bit about some of the lessons I've learned through being involved in this relatively large-scale genomic project here, and as well as through some of my previous work at the NIH and how I think that's affecting healthcare systems like ours and the healthcare systems more generally, as well as society, is for any of us that are practicing anything that really touches genomics. Excuse me. So to start with, I'm going to go into some background information. So many of you may have not heard of Inova previous to this. We're a relatively large healthcare system. As you can see from the map there, we're located in Northern Virginia around the Washington, D.C. region. And we have 6 main hospitals. This includes an independent women and children's hospital. And obviously that goes along with that a relatively large ambulatory healthcare system network. And we serve, as you can see, a lot of patients, about 2 million patient visits a year. And we're especially strong in deliveries. So we have about 20,000 deliveries system-wide per year of infants. About half of those occur on our main campus, where our research institute is located. And our main campus is a complex of different hospital units. Again, it includes a large children's hospital, and we do a lot of deliveries here. Our research institute was started about 5 or 6 years ago, depending on how you do the math. It was started by Dr. John Niederhuber, who's the former head of the National Cancer Institute. So when his presidential appointment as the head of the NCI ended, he came on to be the CEO of the Inova Translational Medicine Institute. And we have all sorts of fancy descriptions and details about what we do, but it really boils down to doing relatively large-scale research on genomics, figuring out how we can use genomics correlated with other biological information and clinical data to make new discoveries about human health and disease, and perhaps just as challengingly, or maybe even more challengingly, how do we take that information and use it in a busy, active healthcare system? And we all know this is a very fast-moving target, and the technologies and the analytic techniques are changing very quickly. Regulation often has a hard time keeping track of how quick things are changing. So this is a big and challenging problem, and one that needs a lot of expertise to address. Our staffing, we have approximately 90 to 100 folks now, not counting all the large collaborations we have, is roughly a third clinical, a third IT, informatic, classic bioinformatic, and about a third lab. And we do both clinical care as well as a lot of research-based applications. These span things from the lab to the bedside and back and forth. I do want to mention briefly that Inova very recently purchased, and actually just about a month and a half ago got the keys to this complex that you're seeing now. This was the former headquarters of ExxonMobil. They've since moved to Texas. This has been bought and is being re-outfitted as the Inova Center for Personalized Health, which will be dedicated in large part to the practice of personalized or precision medicine, and especially genomics. That picture at the top is the governor of the Commonwealth of Virginia, Terry McAuliffe, at the announcement, and you can see the man smiling up at him happily. That's the CEO, who's obviously very excited that we're able to take possession of this property. And again, this is about a 1.2 million square foot complex, and all around there, which is not typical for the Washington, D.C. metropolitan area, is a lot of unused land, and we plan to additionally build other buildings that will also be dedicated to personalized and precision medicine. So we're all very excited about the fact that our healthcare system has really put its money where its mouth is in terms of the practice of this type of clinical care. So I'm going to start going into some of the details about our studies now. So our studies revolve around a number of themes. They involve trio-based whole genome sequencing, meaning we always get sequence from mom, dad, and baby in a significant proportion. We also get sequence and participation from other family members, such as aunts and uncles, grandparents, other siblings, and so on. We generate as much other omics as samples allow at the RNA, epigenetic, protein, and other levels. Some of it general, some of it directed at specific hypotheses. Especially of folks that are affected with certain conditions, we attempt to collect tissue. For example, a baby with a cardiac anomaly that's getting cardiac surgery, we try to get tissue that's being discarded after the surgery to have as part of our biorepository to do functional analyses on, validation studies, and so on. We collect clinical data from our electronic healthcare record system, that's EPIC. We also supplement the electronic healthcare record-based data with study-specific surveys that we send out regularly to our participants. And we bring everything together into a unified database to increase statistical power regardless of the specific question being asked. I'll go into some of what these studies are, but we have a couple relatively large studies. And then as you can see on the right where it says diseases, a number of smaller studies more targeting specific conditions. This gives you a sense of the number of participants that we have enrolled. That's on the left, and on the right, the number of what I call clinical genomes. And so those are genomes that we have on hand with matched clinical data. And as you can see, the numbers for enrolled are larger than the number of clinical genomes. This is about 4 or 5 months out of date because we tend to batch things except for folks that are obviously clinically affected with a condition that we want to analyze genomically. So we batch things and things lag behind a little bit there. But as you heard in the introduction, we're operating currently at around 7,000 to 8,000 genomes or so. And you'll see the numbers, how we accrue things. I'm not going to go into all the studies shown in detail, not even the ones here, but I want to briefly mention these are kind of our 3 main studies. They're also the 3 earliest studies. The first shown in red there is what we call our longitudinal study. And in the next couple slides, we'll go into more details there. But that's a study that's not biased for or against any particular conditions. And we enroll folks about halfway through pregnancy and then follow them prospectively. The preterm birth study, shown in blue, is about half cases, half controls, again, all trio-based whole genome sequencing. That's, as the name implies, aimed at finding the causes, the molecular causes of preterm birth. And we have finished accrual for that, but we're very deep into the analytics there with trying to find the multi-omic and clinical signatures that help us understand and predict preterm birth. And then our smallest study, because of the type of disorder, is shown in green there. And what that is what we call our study of congenital disorders or congenital anomalies. And so these are conditions that are typically manifest early in life, often in the NICU, may not be obvious right away, but we can try to throw N of 1 analyses, multiple pipelines at these questions, at these conditions to try to find genomic answers there. The longitudinal study, which I think is fair to say is our flagship study, and it's honestly what really brought me to ITMI, to Inova, to try to be part of this study, which I really think is groundbreaking and exciting. The idea, again, is that we, according to our theme, we do trio-based whole genome sequencing. We generate other omics, and we take clinical data prospectively on these patients. The original goal was 5,000. Now we're working towards a goal of perhaps 10,000 trio-based families. Again, this is a longitudinal study. We hope that folks will stay enrolled throughout their entire lives and hopefully throughout perhaps even the lives of their children and their children's children. These patients are enrolled about halfway through pregnancy, usually when they enter the second trimester, out of 6 OB offices, all of which deliver at our main hospital here on campus. 4 of the hospitals, or 4 of the practices rather, are standard private care OB practices. One is a high-risk perinatal clinic that's right here in the hospital. And then the other is what's called the Inova Cares Clinic, which is a clinic for the uninsured and underinsured. And we feel strongly that we want to democratize genomics both for clinical care and for research participation. I think one of the issues with genomics and some of these new technologies, and this is true of other types of studies as well, is that they tend to be very biased towards people that are wealthy enough to have the time or the means to be able to take time off from work to participate or to seek out studies. And again, we're trying to really make these studies available to folks regardless of socioeconomic status or any other type questions like that. We collect the types of specimens that are shown there on that 4th bullet point, and then we generate the type of omics that I mentioned earlier, and again that are shown there. And again, we marry this all with clinical data. I know this is a hard slide to see exactly, but this just gives you a rough sense of the accrual. And so we're at about 2,800 families now. Again, the slide is a little bit out of date, but we get between 60 and 80 new families in the longitudinal study per month or so. And like any other study, it can wax and it can wane a little bit, but we're pretty steady there. I don't want to say to my surprise, but we have a terrific group of clinical research associates that are embedded in these various OB practices. And so people, including the obstetricians, the nurses that work with them, and certainly the patients, have been very eager to participate in the study. And I think that's something that before we started this, we honestly didn't really know. Were people going to be interested in this? Would this perhaps interrupt workflows in busy clinical practices and so on? But it seems to be working out very well. Excuse me. One thing I want to mention here is that partly serendipitously, but again, partly intentionally, because we've tried to emphasize this idea that we're being democratic in terms of participation, is that we do have a very diverse population of folks. And so the map is showing you where people in our study were born. We have genome on all these people. Obviously, the people who were born outside the United States came to the United States to have their babies. So all the babies were born here in the Commonwealth of Virginia. Of course, a number of the parents themselves were born in the United States as well. Some of the numbers here are overlapping, but we have especially high numbers in folks of Central American, parts of South American origin, but then people from all over the world, including Africa, as you can see, Asia, Europe, Middle East, and so on. And then on the other side there, of course, is just a simple PCA plot showing for about the first 600 to 700 parents in our studies, just ancestrally where they're from. And I'm not going to go into this point in detail, but of course, having a very diverse cohort is a double-edged sword. If we're trying to understand the causes of a multifactorial or complex condition like preterm birth, it can make things more difficult, where the ideal population might be much more homogeneous. Of course, when we're doing N-of-1 analyses, it makes this a very powerful tool, because we can analyze a specific variant that we find, not only based on the population overall, but that we can look at that allele frequency based on the specific subpopulation. So for example, if someone's of Korean ancestry, we can compare the prevalence of that variant in other folks of that or more related ancestries. And again, I'm not going to go into this, but we do a lot of work on evolutionary genetics in this light and population genomics, all aimed at helping inform us being able to better assign or more intelligently assign pathogenicity or deleteriousness to an allele that we detect. I mentioned the congenital disorder study. I'm just going to come back to that now because that's where some of the cases are going to come from that I'm going to talk to you about. This is mostly a NICU-based study. We have a pretty large NICU here on campus, usually over 100 kids in it. But we also include, other than children in the NICU, other kids with congenital or genetic disorders. Again, some of these are obvious at birth. Some of these, such as developmental delay or immunodeficiencies, might not show up until slightly later. Again, it's a much smaller study because fortunately these conditions aren't as common. But we get a few families a week on average, and we use the same methods I mentioned before, trio-based whole genome sequencing, other omics, all married with clinical data. Another of the things I'm not going to go into detail, but as you can imagine, having the amount and the diversity of the different types of data that we have to have an incredibly robust IT infrastructure. And our leaders on the IT side, Aaron Black and Greg Ely, have done a great deal of work and spent a lot of sleepless nights, them and their teams, making sure that all the data are appropriately secured, that all the data types can be used to talk to each other and can correlate, that we can allow people to use these data sources that we're collaborating with or parts of these data sources, but that we do so in a way that's in accordance with our IRB and that's carefully controlled. That takes a lot of work, again, as you can imagine, and it's something that really enables this genomic engine of ours to run. So with that, I'm going to switch gears a fair bit now and just to go into a few illustrative specific case examples and run to see some of the types of patients we look at. And then I also want to highlight some of the ways that I use the Ingenuity pipelines, which has honestly been a lot of fun and has been very successful. I want to put this in context a little bit. I think it's important to point to the fact, and I'm sure a lot of the people listening to this webinar realize this, but a lot of these Mendelian disorders, that's Gregor Mendel, of course, on that stamp, or these congenital disorders are individually rare and might have only been reported in a couple people in the world or a few families. But in aggregate, they're pretty common. About 5% of live-born infants have these types of conditions. And a lot of these conditions aren't obvious right at birth, might not show up till early adulthood. And it's estimated that 8% or perhaps even higher by age 25 have these types of conditions. So again, while these are individually rare in aggregate, they're very common, and so it's important to be able to recognize, to diagnose, and therefore be able to better treat patients with these conditions. These images, and I'll go into a few of these in detail as part of some of the examples, show some of the types of conditions that we've studied. And so these include conditions that affect the skeleton, different organ systems, the brain, and other organ systems. And that pie chart at the bottom is showing you diagnoses in our longitudinal cohort. That's the cohort I talked about before. This is diagnoses at about 1 year of age. And so in blue, those are most of the kids who are healthy, right, and fortunately don't have diseases. If they're diagnosed with a condition, such as diagnoses of developmental delay or asthma or other conditions, we can start to build cohorts around those and look at those genomically to see if we can detect answers there, either on an N of 1 or kind of a combined basis. And then in colors, those are the different conditions that look congenital that have showed up in our population just by 1 year of age. And I very, in a quick and dirty way, kind of assign different organ systems that they've been affected, but of course some of those conditions affect multiple organ systems. And again, what was perhaps surprising to me, though, is that we get the expected proportion of health and disease within our longitudinal cohort, meaning it really is a very good control cohort, and we use it as a control in a lot of situations. So we see a lot of the rare conditions, not all of them, of course, because of the size of the cohort, but we do see those, and we see a lot of kids who are fortunately healthy. I want to make this point because I think it's a very important point. I think a lot of us on the call as people involved in genomics or one way or another, we realize the importance of being able to diagnose these kids, but I want to underscore that point with some concrete data. And I know these data because I spent a lot of sleepless nights working on this. So currently, if you look pretty strictly, as of a few weeks ago, there are a little over 3,000 genes known to be involved in Mendelian disorders. Of course, there are many more Mendelian disorders than this. Not the causes of all of them haven't been discovered, and some genes might have multiple, sometimes even more than a dozen different allelic conditions. And so I went through and I counted one by one to think about what could be done different. Would medical care be different, more tailored, if we knew the underlying molecular cause? And in almost half the time, you would do different interventions with molecular causes. And I think this actually downplays things a lot. So this wouldn't include things or conditions where it would be beneficial to have the underlying molecular diagnosis, where sometimes that information might not lead to something dramatic in terms of differences in treatment, but certainly could affect things like medical decision-making, information about prognosis, certainly providing knowledge to patients and families so they're not going through these endless kind of diagnostic odysseys, as the term goes these days. Of course, a lot of these conditions, these Mendelian conditions, manifest in childhood, so it's not surprising that most of the time, almost 96% of the time, the interventions are done during childhood. And I keep a database of these, which is still housed at the National Institutes of Health through the NHGRI. I update this just about daily, and then every 1 to 2 months, a new update is pushed through to the website. So if you're interested in a certain condition and knowing would something be done differently if the molecular cause were known, one can go to this database and find this, as well as links to other resources such as dbSNP, such as ClinVar, and then specific clinical information about those. The other point I'd like to emphasize here is that a lot through the successes of things like genomic sequencing and related technologies, that a tremendous wave of discovery has happened. So I looked at the last 2 years, for example, and on average over the last 2 years, over 12 new disease genes per month were discovered as being involved in human conditions, which is, as you can imagine, a lot of genes. And this doesn't even count new allelic conditions that have been discovered with previously implicated genes. And if you look at the x-axis here, I think what's really impressive is that the vast majority were done using some sort of genomic technique, either exomes or genomes. And a lot of them were done just by using, for example, if you can see that first tick line on the x-axis, just by exomes alone. So doing exomes on maybe a handful of patients or a couple of families, and not having to do long preliminary analyses like linkage analysis or even homozygosity mapping or so on, really changed the game and the speed at which things proceed. Very different, obviously, than how things were a decade ago. Unfortunately, I think current practice still tends to be this, and I know this is hard to see, so I'll just explain what this is. This is a timeline for a child born in our NICU who had kind of a confusing mix of different conditions, and each box there shows a different test that was done to try to figure out what was going on with the child. Some of these were traditional genetic tests like karyotype and microarray or sequencing panels. Some of them were biochemical tests. Some of these were radiologic studies. Some of these were clinical tests, like having a good pediatric ophthalmologist try to look at the child and see what was going on. And so what ends up happening is that often we do this test after test after test, this kind of death by 1,000 cuts approach, until someone finally throws up their hands and says, okay, fine, we're going to do an exome or a genome, and we get the answer that way. And I think this is problematic for a couple of reasons. One is that obviously we don't get answers to the patients and the families as soon as we could. Secondly, this ends up therefore hampering clinical care, right? So we can't do more tailored, as I showed you on the previous slides, more tailored, more specific care that's often beneficial. And then third, this ends up being a much more expensive way to practice medicine, right? We're doing lots of tests that are individually not as cheap, not as expensive, but in aggregate are expensive. And then finally we add on a test at the end. And I know this is controversial, but I would advocate for doing some of these genomic testings, some of these genomic applications earlier in the process. And I think we've shown some good evidence that this is a wise way to proceed, and certainly other groups are looking at this as well, such as groups in Toronto, such as Stephen Kingsmore, who's recently gone to San Diego, that we should apply these techniques earlier in a lot of situations. And as an example of this, this was a child, as you can see, born in our NICU. This child had pretty massive hepatosplenomegaly, and so this imaging here is showing you that this child had a very large liver and a very large spleen. And at first it wasn't clear what this child had. It was thought that it might be some kind of vascular or anatomic anomaly that was basically causing blood to back up and make these organs enlarge. And we did genomics on this child and found out pretty quickly that the child had Niemann-Pick type C disease. And I remember very vividly, I was at ASHG in California, and I got a call from one of the bioinformaticists asking, could this be Niemann-Pick? And on my phone, I'm able to get the electronic healthcare record and look. And I thought, yeah, and I looked and I said, yeah, absolutely, this kid does. This was something that was on our differential, but we didn't know for sure. And so the kid had a clinical panel that was out at the same time at one of the commercial labs, and this gene happened to be on it, and I called up that lab immediately and said that we had found this on a research basis. Can they please confirm this very, very quickly, because we need this information for clinical care? And so we were able to do that and get them to rush it to get the CLIA-validated answer very quick. Shown there is our Sanger validation of it to make sure it was real. And we got that turned around very quickly, and then got the answer to the kid. So I got the call on Friday. We got the Sanger turned around in the next couple of days, and the kid by Monday or Tuesday in with a biochemical geneticist in discussing with the transplant surgeon and others. And all this was directed very fortuitously through Denny Porter, who's one of the world's experts on this condition. He's the clinical director of NICHD. And it was a very rewarding thing to know that, by finding this answer, we could have the world's experts on these conditions talking to each other to make sure that the child got the best possible care. For example, he was able to get IV cyclodextrin that was aimed to help his liver hold over until transplant. He's since had transplant and is doing well from that. So I just want to show you kind of some of the examples. I want to emphasize that we use a lot of different pipelines. I think all pipelines have different pluses and minuses. So I'm going to show you some cases using the Ingenuity pipelines. I will want to be clear, though, that some of these were found through other pipelines first, but I want to show you how easy it is to use the Ingenuity pipeline. And I put some of these cases into their pipeline to show kind of how easy it is to use. And again, as a way of disclosure, I'm a medical geneticist. I'm not a bioinformaticist, so I kind of dabble a little bit, but I would not call myself an expert. But for someone like me, this works amazingly well. It lets me solve cases really quickly. It lets me, when cases I haven't solved, kind of noodle around with them and try to look at new candidate genes or new genes that have just been published, and it's been tremendously helpful. So in this particular case, this case of Niemann-Pick type C, I uploaded the data, and that took about a minute. I plugged in a few quick things, like said this is a genetic disease, not cancer, and just to kind of do the standard analysis, and then I pressed run, and I was sitting there using through a secured connection on my laptop at home. My wife was next to me watching, I think, NCIS or something. I got up to get a drink of orange juice. I sat back down. I already had the email saying that your analysis is ready, this is the little email, and I got that about a minute after I submitted it. And then, as you can see from this, I just went in and took another 30 seconds to load, and I immediately had, from this trio-based whole genome sequencing data, I immediately had a single answer, which I know is correct because I did a kind of a true positive case. And this is just breathtaking to me. I know I come across as doing an ad for ingenuity now, but it's really a powerful tool. As one of the bioinformaticists I work with said, it's just not fair that we have these tools that enable us to provide answers so quickly. It's really tremendously rewarding. And again, I love the fact that I, as a pediatric geneticist, as a clinician, that I can go into these and use these tools and do things with a clinical mind, and I don't have to wait necessarily for the bioinformaticist to do some of the analyses that they do. Obviously, those can be very important and powerful as well. This is another case. If you want to read more about this, here's the citation there. So this was a child that was, again, born in our NICU, was born a little bit early, was IUGR, which means the child was smaller, had intrauterine growth restriction when the mom was pregnant, and then had this vague cluster of kind of confusing medical conditions. So this is a 3D reconstruction of the skull. The skull shape is a little different than usual. The profile, you can see the forehead has a little kind of what's called frontal bossing, so a little more prominent than usual. And in the bottom view, we're looking at the back of the skull. And what we're seeing there is that we're seeing the sagittal suture, that suture going down the bottom, is connected, and it shouldn't be. And so you have to have surgery to separate that. And the child also had this kind of, wasn't growing terrifically, seemed to have some renal, some kidney differences, maybe some endocrine differences, but not really clear at all what was going on. And as a lot of these kids do, he had a pretty extensive workup and a bunch of different tests, both molecular tests, biochemical tests that didn't show the answer. So we did a whole genome on the folks shown here, so the kid shown at the bottom, and then also that's the child, and also the other members of the family, and everybody else in the family was healthy. Didn't seem to have any signs of disease or really anything similar at all to the child. So we did whole genome sequencing. We found a mutation in this child. We found the same mutation in these other folks. These other folks were unaffected. And by instituting a specific imprinting type pipeline, we were able to figure out that this mutation was the cause, and otherwise it would have been overlooked. And so you only get this condition if you have the mutation and if you get the mutation from the mother, right? Because if you get the mutation from the dad, the gene is, and this is a bad way to put it, but not active or kind of turned off. And what's interesting is when we went back and told the family about this, the grandfather there at the top said, oh yeah, I had this distant relative, and I never thought about this, but he had the same thing, or she rather, had the same thing. It reminded me of what my grandson had, and the inheritance was such, even though that person has long since passed away, that it could have been the same condition. And I've since had conversations with the other members of this family for family planning, things like pre-implantation genetic diagnosis, now that they know they carry this mutation. I think the other thing that was clinically dramatic about this is that when we figured this out, only about 10 or 12 other folks in the world had this condition. And all the other folks in the world had pretty frank and pretty severe adrenal hypoplasia, so underdevelopment of the adrenal glands. And so in these other kids, it was severe enough that the kids were getting adrenal replacement. This child, we figured out, one of my bioinformaticists told me that we had this figured out, and I realized in looking at the medical record that this child was about to go into surgery. And so knowing that he was at high risk of an adrenal crisis, we were able to quickly get on the phone, talk to the anesthesiologist, talk to the surgeons and the other folks taking care of him to make sure that they did a very simple thing, just gave him stress dose steroids prior to surgery because his adrenal glands don't make the steroids that they're supposed to. And so I don't know if this is a life-saving thing, but it certainly could have been, and certainly very dramatic and eye-opening experience to me about the power of some of these methods and some of these analyses. So another case, this was a child, again, these are all kids from our NICU. This is neuroimaging. These arrows are showing you that this child had these kind of like stroke-like episodes during infancy, also had severe dermatologic conditions, a lot of skin missing on the scalp, but not clear what was going on. These are at the bottom there in this figure. This is our patient, and then some other patients shown above. This child did not have severe limb hypoplasia. She had maybe kind of hypoplastic toenails. She had scalp lesions like some of these folks had that are shown there. But We found this, and then in collaboration with a group at University of British Columbia led by Milan Patel, and then our collaborators at Institutes for Systems Biology led by Jared Roach and Gustavo Glusman, their team, they had 4 other families with mutations in this same gene. And so this is a brand new cause of disease, and now the 5th cause of what's called Adams-Oliver syndrome. And it's amazing, I guess the emphasis is it's amazing how quickly we can go from answers to the new answer for a genetic condition, a new published answer. And actually, since this was published, our other collaborators who continue to work on this condition have found a 6th gene. So now there's 6 genes that can explain Adams-Oliver. Again, a really fast-moving field here. And these are some of the collaborators. I think this is funny. I think only a geneticist would be amused by this. But this is from their press release about this. This is a dominant condition, and we think it's very funny that this is showing you consanguinity or recessive condition. But they are terrific genomicists. That's Jared Roach and Gustavo Glusman, certainly some of the leaders in this field. So just another case. This is a child, as you can see from the arrows, born with vertebral anomalies, had to get surgery, and born with a single kidney. Otherwise doing pretty well. And this is the child when she came in and saw us. And again, the really amazing thing to me was that this was a de novo mutation in EP300. And some of you guys might know EP300 mutations are supposed to give you Rubinstein-Taybi syndrome, which isn't supposed to have birth defects, which has a characteristic facial appearance, and this almost pathognomonic appearance to the thumbs and the big toes. And this child, I would argue, doesn't have those. And I think it's an example, again, of how using genomics on a broader, much more hypothesis-free way is allowing us to rewrite the textbooks about what happens, what manifests from mutations. And a lot of times, this is a very humbling thing because what we thought we knew that was caused by mutations in certain genes is just not true at all. We're really stretching the phenotypes that we're getting. And I think our view of this will be very different 10 years from now when many more people are sequenced than they are now. Another example of this was a child with Ebstein's anomaly, and that figure showing you this is an anomaly affecting one of the valves of the heart. The child had Ebstein's anomaly, as did the dad. The child also had other cardiac anomalies. And there aren't really published genes that give you kind of isolated Ebstein's anomaly. There are that give you Ebstein's anomaly with other things, but not that. But again, I wanted to show the power of the QIAGEN pipeline here. So this is just some screenshots from using the pipeline. It's incredibly user-friendly. And I feel like I'm good at a few things. I'll admit that one of the things I'm not good at is computer programming, and I'm not the most tech-savvy person, I have to admit. But even for me, this works tremendously well. We just kind of pulled over, dropped and dragged the genomic files from there, and that's what that screenshot's just showing you. And then I just typed in one clinical thing. I just typed in Ebstein's anomaly and put it in there. And again, I hit analyze, got up for 30 seconds or so to get a snack, sat back down, and I had the right answer, which was a mutation in MYH7 in this case that both the child and the dad had. Again, I think this is, it's not that dramatic because we know that MYH7 mutations can cause Ebstein's anomaly. But as far as I could tell, and I looked at the literature, when I saw this child before we had the answer pretty quickly, Ebstein's anomaly is also supposed to go, MYH7 mutations are also supposed to go along with other cardiac findings like ventricular non-compaction, not just isolated Ebstein's anomaly like the child had. So again, we're really stretching the phenotypic range that we get from a lot of these conditions. And this is from the consult note, and this is the original consult note saying, it just doesn't make sense. MYH7 couldn't be involved. And of course, we found an MYH7 mutation in this kid. I often, when I present this case, I kind of joke around about who the dopey consultant was, and it was me, and again, this was an eye-opening experience. And that's my boss, by the way, behind me, John Niederhuber, the ex-head of NCI, looking skeptically at me as I try to explain something. That's often how he looks at me. All right, so I hope these cases have been helpful to illustrate some of the types of patients we have, as well as about the type of data set that we use to derive this. And again, I really enjoy using the Ingenuity variant tools to look at this, and they've been tremendously powerful, as you can see. I continue to look at a lot of cases this way, including some cases that honestly I don't think are going to be that easy to solve on an N of 1 basis. For example, we have over a dozen kids with gastroschisis, which is a congenital anomaly affecting the abdominal wall. I don't think we're going to have a kind of an obvious, easy Mendelian answer, but this allows us to aggregate these cases and look for common patterns using these tools. I haven't found anything obvious yet, though we still have I have some leads on gastroschisis. But again, using these tools is really fun, and when there's a new gene published or some new insight, we can go back and reanalyze and see if we get new answers that way. So again, if you'll indulge me, I'm going to just try to communicate some lessons learned. And the first, and hopefully this has come across pretty clearly already, is this idea that the diagnostic paradigm has shifted. And so we used to be kind of in this diagnostic paradigm. This is a show House, some of you guys might recognize. I believe this is still in reruns, though perhaps there isn't in primary production anymore. But kind of the House modality of testing, right, is that someone would come in with a confusing type of condition or something we really didn't know what the cause of it was, and we'd do one test and that would be wrong, and we'd scratch our heads and do another test and that would be wrong, and we'd scratch our heads and have all sorts of looking in databases and textbooks and calling our friends, and in the case of House, perhaps going and having an affair or something, and finally we get an answer. And things have changed, and this is, to me as a physician, this is very humbling, where it's no longer us necessarily as the physician selecting the test, but rather generating a lot of omic and clinical data. And my role now is rather to help sort or help kind of clinically think about these data rather than ordering this isolated test or that isolated test. And I think this is it's a tough pill to swallow for a lot of physicians, a lot of folks who are used to being kind of the sole proprietor of the answers. The other thing I want to emphasize, and I know we're on a webinar now, so I can't ask folks kind of for a show of hands, but there's this sense about where we are on this curve of genomic or precision medicine or personalized medicine. And I know different people have different views about this, and I won't pick on anybody to ask where they are, but I do think that it's important to manage perceptions, is that we're very early in the game. And so I like to say, and some of the folks I work with like to say, we're at the Wright brothers stage of this, or perhaps we're at the the Model T stage of this, is that we're very early in the game. We have a great deal to learn about how a single variant affects a person's health, especially variants that are outside the traditional exome or the exons. And I think we have even more work to do, tremendous work to do, to understand how these variants affect health not only alone, but in concert with each other and in combination with other non-genetic factors. And again, how these help explain and help predict health. We have a lot of work to do. And I don't say that just as kind of as lobbying towards job security, but I think it's important that to the public that we don't overemphasize the ability of personalized or precision medicine to provide answers, but that we describe things carefully and conservatively. The other thing along these lines is that we really do need to provide help and guidance. That's just that picture is just a picture of a biochemical type pathways. Obviously, genomics has a lot to do with that and is very complex. And that we know that clinicians, nurses, doctors, pharmacists, others are already incredibly taxed and have a lot of work to do with the way that the clinical system is set up. They're constantly being asked to see more patients and in less time. And I think, again, the onus is on us, if you allow me to be up on the soapbox, to provide tools for these folks, to provide guidance for these folks, so that we're really working with them and that we're making their life better and easier by delivering this genomic information, not more complex, not more challenging, not harder, so that we're providing more answers rather than more questions and difficulties. Our research institute works very hard in that realm, and we have a kind of a living laboratory in that way in terms of working alongside a lot of the clinicians. And it's been a very eye and mind-opening experience, seeing what can help them, what doesn't help them, and what still needs a lot of work. For example, one of the things that we do that I'm proud of, it's very simple. It's not nearly as complex as a lot of things, but we simply have a very easy chat line so that if folks want to get in touch with us about any question about genetics or genomics or what tube do I put this in or how do I make sense of this variant of uncertain significance, we have just a simple chat so that folks can input information. It comes to either my phone immediately, or if I'm online, it comes to my computer desktop. It's also linked to the phones and desktops of a couple of my genetic counselors, a bioinformaticist, one of my postdocs as well, so we can try to get answers for these folks. And with that, I think the most important line, or the most important lesson rather, is that it really is an amazingly fun time to be a geneticist, or perhaps we should call ourselves a genomicist. You know, to be able to generate these types of data sets, to have these types of tools at our fingertips is really quite wonderful, quite astounding. I don't think any of us would have guessed that we'd be able to do what we can do, 10 or 15 years ago. I think 10 or 15 years from now, things again will be tremendously difficult. Will be tremendously difficult, but will also be tremendously rewarding. I think we have a ways to go, but we certainly have a lot of material and a lot of tools to get there. I'm certainly excited about that. I'd like to thank you guys very much for your time and your patience and hopefully your interest. I'd like to acknowledge all the folks listed here. For the sake of time, I'm not going to go through any particular names except for the heads of the Inova Translational Medicine Institute, or ITMI, John Niederhuber, again, the former director of NCI, Joe Vockley, who's our current Chief Scientific Officer and our Chief Operations Officer, who also previously, many of you guys may know, who ran the Cancer Genome Atlas project out of NIH and NCI. And again, I'd really like to thank QIAGEN and Ingenuity for the opportunity to present. I'm going to end now, and I'm happy to answer any questions at this point, or take questions offline if folks want to contact the administrators. Thanks very much.
Thank you, Dr. Solomon, for that informative presentation. It is time for Q&A. So if you have a question you'd like to ask Dr. Solomon, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on the screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how is the genomic work you do funded?
Thanks. Yes, I think this is a great question, and I often put this in here to make sure that this comes up, and I think this partly speaks about why I'm talking to you guys now. So our work is all internally funded, meaning our healthcare system believes very strongly in the future of genomics and personalized and precision medicine. So they brought John Niederhuber here to start this institute and to invest very heavily in genomics. Obviously, the good side of that is that we have healthy funding. The downside of that, perhaps one might say, is that we're not required, such as an NIH-funded study, to put things in the public domain. However, we feel it's our responsibility as researchers, bioethically, to help others find answers. And part of what we've done is collaborated with Ingenuity and QIAGEN to try to make these data available for things, for example, to be used as a controlled database so that folks get access to that. And we feel that's the best way that we can secure the data, keep it safe and protected in the way that our IRB requires and the folks who participate in our study, the way they consented to it, but also, again, helps fulfill our ethical responsibility to be part of the larger genomic community.
The next person says, I believe you just answered my question, but I'll ask anyhow. Your lab still uses Sanger sequencing methods to verify exome data?
Yeah, so it's a very great question. So let me answer this in a couple different ways, or at a couple different angles. So first, we do genomes, not exomes. I don't think we've generated many exomes at all, maybe a few just for kind of quality control purposes, but it's all genomes. So yeah, so what we do is we still do orthogonal validation of findings before we return them to patients. And I know there's a lot of discussion at this point about statistically showing that next generation sequencing data, especially if they have a certain depth, and there are questions about the need for orthogonal sequencing. So right now we try to be very careful, and when we're returning things to patients, we do still do Sanger sequencing from the original research samples, and then we typically also require a separate sample to do CLIA validation. Remember that the stuff that I talked to you guys about is all done on a research basis. So we are still doing Sanger or the equivalent validation, some other type of validation on top of this. I do recognize, I think it's a great question because I think this is a fast-moving field. And for example, I'm sure you guys have heard talks and seen data where, for example, the Sanger data is actually less trustworthy than the original next-generation sequencing data. But I think that we and others are in many ways trying to move a little bit cautiously and conservatively until more evidence comes out there. But it's as you guys all know, it's a very fast-moving field. It's a great question.
Why did you choose to do whole genome sequencing instead of exome sequencing?
Yeah, so another good and important question. I think this raises a number of points. So I think a lot of us believe that things are moving towards whole genome rather than whole exome, and kind of when that turnover happens, I think is none of us are exactly clear about. But the idea is that if we wanted to generate a data set that would be useful for hopefully a lot of time to come, genome was the way to go. Obviously, there's downsides to that, including the expense and the turnaround time and things like that. But we wanted to generate a data set that was applicable, hopefully for the longer term, as well as, we're very interested. I talked a lot about Mendelian diseases, and we know that a lot, but certainly not all of the answers are in the exome for these Mendelian diseases. But a lot of the work that we do is, of course, related to complex diseases, things like preterm birth and things like conditions that we don't expect necessarily to have be due to a single hit from one mutation in the exon. So having the genome enables us to look, of course, outside the exons. And then the other thing along those lines is that we do generate, for example, a fair bit of RNA-based and epigenetic data. So hopefully it'll enable us to correlate some of those non-exonic variants with some of those data sets as well.
In the longitudinal study, how frequently do you follow up with the participants, and what level of response rates have you seen?
Yeah, so it's again a good and important question. So we collect information from our electronic healthcare record on these folks. The other thing that we do is that every 6 months we have web-based surveys that go out to them. Mostly they answer them through the web, through email, but they can also answer them through traditional mail or through the phone. And we actually have a tremendous rate of return, well over 80%, in some cases over 90% of return on those data. And I think that speaks to having a dedicated and very good clinical team, primarily clinical nurses, whose main job is it to make sure that we have folks in our studies that are continuing to answer questionnaires and continuing to be invested. We try to be careful. We try not to be pushy and do things that we certainly never do anything that the IRB or the consent forms don't allow. But I think the other point that it speaks to is that folks are certainly very eager for these genomics and for research participation. It doesn't cost them anything to do this other than time. I think they're very eager and kind of like being part of this big study.
Your presentation has focused on individual patient cases. Has your group or have other groups done any larger analyses using the data set?
Yeah, so another good question. So we recently published, for example, on Genetics and Medicine, kind of a smallish, but I think nice analysis where we looked at 163 genes corresponding to all the conditions that are screened for in the United States in newborn screening programs, in the blood-based newborn screening program, or are being planned to be initiated in the United States, or by now actually have been initiated in a lot of cases, such as some of the types of immunodeficiencies that have been added. And so we've done these ones like that are, I guess one could call these combined N of 1 analyses, right, because we're looking at gene sets there. And we're doing similar analyses of combined data sets like that, looking at other groups that are kind of logical to look at, ones having to do with immunodeficiencies, ones having to do with hearing loss. Dale Bodie, one of our bioinformaticists, also published an analysis looking at genes from the cosmic gene census and so on. And then we're very busy, and again, I know I didn't really get to that in any of the slides, but we're very busy looking at larger analyses, especially of things like preterm birth, where we have some of these cohorts, and then smaller ones too, where just kind of naturally we've had groups of patients with things like gastroschisis, I mentioned as an example, where we have a dozen cases. And as the data set matures, meaning as the kids in the study get older, we expect to see more of these conditions that don't pop up early in life. So we're now starting to get diagnoses of kids with developmental delay, things like asthma, things like autism, frequent infections, and so we can kind of build these cohorts out of these as we're starting to see some of these diagnoses being made.
I would like to once again thank Dr. Solomon for his presentation. Do you have any final comments?
No, I'd just like to thank everybody for their time. I'd like to welcome folks to contact me if they have questions about the data set at all. And again, I think I'd just like to leave on a very positive note that this is a really just terrific field to be in, and I think it will continue to be even more terrific as data sets grow and as collaborations grow and as more data are available. Thank you.
Thank you again, Dr. Solomon. I would also like to thank our sponsor, QIAGEN, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 12, 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye.